Okay, hello everybody, and welcome to today's seminar for machine learning for protein engineering. It is my deepest pleasure to welcome fifth year Tianhao Yu. He is coming to us from UIUC, part of Huimin Zhao's group, and today he's going to be presenting his newest work on enzyme function prediction using contrastive learning. So whenever you're ready, Tianhao, we are ready to hear your talk. Thank you so much.
Thanks, Mac. Thanks for the nice introduction. And then thank you for all the organizer for inviting me to give a talk at this seminar. I'm really excited. So today I'll be sharing our work. Title is Enzyme Function Prediction Using Contrastive Learning. And my name is Tianhao. My advisor, Huimin Zhao, as Mac introduced. So this work was published last year in May. Oh, sorry, in March, in late March. And yeah, so let's get into it. First, I guess at this seminar, I don't need to emphasize too much on our motivation to focus on enzyme. Enzymes are great because they are nature's biocatalysis. So compared with traditional organic chemical catalyst, enzymes are have two unique ability. It's very selective, and then also it promotes sustainability or achieve great chemistry. It's selective in terms of substrate selectivity, regional selectivity, and the stereo selectivities. So the green chemistry can be achieved because enzymes reactions can be performed in aqueous solution as opposed to some traditional chemical catalysts has to be in harsh or extreme conditions. And in the industry, many of these commercial products are already partially or fully synthesized by enzymatic pathway. It spans across like drugs, food additives, and many other fine chemicals. So particularly for enzymes, there is one barrier that hinders biologists to unleash the full potential of enzymes. Because the number of available protein sequences are increasing. However, we don't have a very robust way to functionally annotate those. So we can take out the statistic I pull out from UniProt database, one of the largest protein database. So the first graph shows the unreviewed portion of the UniProt. It has hundreds of millions of sequences by the year of 2020. However, the reviewed portion, if we take a look, it's the scale of the Y scale is only hundreds of thousands. So it's only like 0.5% of all the UniProt database has been expertly reviewed. If we look at the growth rate, these are just some time step I took in April 2 years ago and 1 year ago. So the unreviewed portion has grown by 15 million sequences, but the reviewed portion only grow by 2,000. Therefore, it leads to a question that we cannot rely on human experts to review everything, and we certainly cannot rely on experimental efforts to annotate every single sequences. Therefore, we do need a automatic tool, a computational tool to annotate these raw sequences with high robustity and then high accuracy. So some background that people have been using the annotation methods. The community has actually a long history of computational annotation tools. So one of the very gold standard and effective yet old method is using bioinformatic tools. It could be like BLASTp, the Markov model, motif detection, or domain detection, and many other versions of different bioinformatic tools. So the underlying logic is really to do some sort of alignment, finding similarities between the query sequences and a large database with annotated sequences. So once the bioinformatic tools can find a homolog or a close relative in the annotated database, we can transfer the label accordingly. However, according to a Nature study in 2018 by Price and coworkers, a third of the sequences, like a third of the raw amino acid sequences can still not be annotated automatically using the collective of all different computational tools available. So very recently, people have turned their attention to using machine learning to tackle this problem. Essentially, machine learning, people try to train on the large annotated database and then to inference the predictive function directly. So the output of the function annotation can be in various different format. It could be just the name of the enzyme. For example, keto reductase. It'll tells you what the enzyme can, what reaction the enzyme can catalyze. It could be an EC number. Which is a numerical scheme to classify enzyme functions. It could be gene ontology terms and many different ways. The problem with recent machine learning methods developed is that the accuracy is actually a bit lower than the bioinformatic tools. The standalone of a machine learning performance is slower than BLASTp, for example. And then because the enzyme annotations are known to be very biased data set. Therefore, some of the understudied functions lack of data to support the training process, and they perform very poorly on the understudied portion of the functions. So for our work particularly, we focus on enzymes. So EC number are the labels we choose to characterize the enzyme functions. So to give you a little bit background on what EC numbers are, essentially EC numbers in short is the enzyme commission number. It's a hierarchical structure as a numerical scheme to classify enzyme functions. Now, it has 4 digits. So let's just take EC number 1.2.3.4 as an example. At the very top level, it has 7 different EC numbers. Here, just take an example. EC number 1 is the oxidoreductases. So all the oxidoreductases will have EC numbers beginning with the first digit 1. And then at the second level, it has further to specify what type of oxidoreductase are. For example, EC number 1.2 will be those oxidoreductase that acting on aldehyde or oxo group as a donor. And then the third level, furthermore specify what the enzyme can do. For example, 1.2.3 will be using oxygen X acceptor. And finally, down to the last digit, the 4th digit, we can have a more focused and more defined enzyme name and their reaction scheme. For example, this one will be a oxalate oxidase. Now, the primary goal or the initial goal of our work is really to distinguish the enzyme promiscuity. So enzyme promiscuity as a term, it means that enzymes sometimes can perform different reactions, and different reactions can mean differently. It actually be defined differently in different literature. It could be mean from substrate promiscuity, meaning that a reaction can perform reactions on different substrate. For example, both of these are alcohol, but it's a slightly different alcohol. But more widely, promiscuity also mean a reaction can do different reactivity or different reactions. Now, these two are just hypothetical. Say if say one enzyme can perform both of these reactions, then they are reaction-wise promiscuous. So in our case, we are using EC number to define promiscuity. So in our study, we basically define promiscuous enzyme as the enzymes with more than 1 EC number associated with it. So if it has 2 distinct EC number down to the 4th digit, then we'll say this enzyme is promiscuous. So after the background, we can review a little bit of the previous work. Mostly there are 2 state-of-the-art models for predicting EC numbers of the enzyme sequences. The first one is called DeepEC. This one is published in 2019 in PNAS by Liu and co-workers. So their method was using 3 independent convolution neural networks combined with a bioinformatic tool called Diamond. Essentially, the way their model worked from left to right is that they first embed the enzyme sequences into one-hot encoding, and then it has been fed through 3 different independent CNNs. The first one will determine if it's an enzyme or not enzyme. The second one will determine the first 3 digits of EC numbers. And then the last one is responsible to predict the last EC number, actually up to the last EC number, so all 4 EC numbers. And then there will be they'll compare the result of the 3 different CNNs. If all of those 3 results aligns, meaning that the result doesn't contradict with each other, the result is compatible, then they'll just use the CNN's result as the output. If not, or if any of the CNNs cannot give a prediction, they'll turn to the homolog analysis using Diamond to do a homolog search, and then use the bioinformatic tools to give the EC number. Now, more recently, there is another work called Protein-Infer. This was published by Sanderson and co-workers in 2023 in eLife. So essentially, Protein-Infer is a dilated convolutionary architecture for protein function prediction. The amino acids are first also being one-hot encoded and then pass through a series of convolution implemented with residual blocks. And then basically the futures will increasingly dilated, allow the top residue layer of the network to kind of come up with a meaningful representation of the protein sequence itself. And on the right is a performance that I got from their paper. As you can see, that the combined CNN with BLAST, highlighted by the light green color, as the best performance, and then the red one is BLAST only. You can see it's actually pretty good. And compared with the single CNN and ensemble CNN, BLAST only is very compatible. So I basically wasn't lying, saying a lot of recent machine learning methods are not as accurate as a gold standard bioinformatic tools. So for us, when we first approached this problem, I begin by using a very naive approach, just to probe a baseline and then see what a problem is. The most intuitive way to do this is just to input a amino acid sequence, get a representation somehow, and then feed through like a MLP, and then use a module label classification to get EC numbers. Now, the major problem of these is with the data balancing issue. So on the right, the histogram illustrates the frequency distribution of the EC numbers across the data set. So essentially, I pull out all the enzyme sequences from UniProt database. To be specific, is the human reviewed portion, also named as SwissProt database. In total, there are more than 20,000 protein sequences, enzyme sequences, and then covering more than 5,000 different EC numbers. However, we can see thousands of these EC numbers on the left pick are, like most of these are pretty rare. They only occurs single-digit times across all this 20,000 possible sequences. So the major problem here, or the major issue here, is we're dealing with a very, very biased data set. So we want to somehow change our model to be able to handle the imbalanced data set. So in order to handle the data imbalance issue, we turn our attention to contrastive learning. So if you're not familiar with contrastive learning, I'll just give a little bit of introduction on contrastive learning. If you are, you can take a 10-minute brief nap. So essentially, the main difference between contrastive learning is that they don't learn the label directly, so they actually learn the similarity between the input features. So through the training process, let's just take a dog and cat example to illustrate how our model works. Essentially for each of the reference picture, let's see if I can get a little more. So for each of the anchor sequences, which is a reference training example, we'll sample another sample from the training data set with the same label as the anchor, and we'll name that sample a positive sample. For example, in this case, we have an anchor which appears to be a dog. So we sample another dog images and then pair them together, call them a positive pair. And then simultaneously, we'll do the reverse, which is, okay. We'll do the reverse, which is sample a negative pairs. So in this case, if an anchor is a dog, then we'll sample for a cat. And then we'll call this a negative pair and a negative sample. So after we have these triplets, the anchor, positive, and negative inputs, we'll be feeding all three to the same model, but three different instances. And each of the anchor, positive, and negative go to each individual instances. And after, and then the output feature was fed to like the triplet loss function. Which is detailed here, is essentially the distance of between anchor and positive minus the distance between anchor and negative plus a constant. So to minimize this loss function, how the contrastive learning work is by minimizing the loss function, we want to minimize the distance between anchor and positive, but then we want to maximize this term. So we want to maximize the distance between anchor and negative. So after training, we would be expecting to see something like this. So all the dogs will be clustered together, all the cats will cluster at a different position. So if we have another query picture, after training, we wanted to infer what's the label of this query. We can just put it on the space, and then if it's closer to the dog, then we say, oh, this is more likely to be a dog. If it's closer to the cat, we say it's a cat. So in short, that's how a triplet loss-based contrastive learning works. And that's exactly what we do. Instead of cat and dog pictures, we just swap those with amino acid sequences, and the cat and dog label will just be EC numbers. So we developed this model, we name it CLEAN, in short for contrastive learning enabled enzyme annotation methods. So we will sample, at the first step, we will sample a pair of sequences with the same EC number according to the anchor sequences. For example, both of them are for EC number 1.1.1.1. And simultaneously, based on this anchor sequence, we'll also sample a negative sequence. In this case, it's 1.1.1.2. And then this sequence will first go through a language model embedding. The one we chose is ESM1B. After the initial feature of the language model, we will go through a MLP, and then the output feature will be used for the triplet marginal loss. The goal, again, for the loss was to minimize the distance between these two sequences with the same EC number. At the same time, we want to maximize distance between the sequences of the anchor and negative. So once the model has been trained, we'll be expecting to see that all the sequences with the same EC number should clustering together distance-wise. And then with a different EC number, will be separated far apart. So now if we have a query sequence, we can just put on the space and then say, oh, this query sequence is very close to 1.1.1. Then we'll say, oh, this query sequence can be, will be labeled as this EC number. Now there's a trick on. I just saw there's one question in the chat saying what data are DPC and protein for train on. So both of these models are trained on UniProt. So basically the same database I use, also UniProt. Yeah, I'll just continue. And then one catch here is that during the training, we also employed a technique or a trick. It's called sample the hard negatives. The underlying logic is that not all negative samples are equal for the model's point of view. So we need to prioritize those challenging negative samples for the model as opposed to very easy or trivial negative samples. So if we take a look again at this loss function, if the features between the anchor and negatives are already very different, then this term is already being maximized, so it doesn't do anything to the loss function. That's why we want to choose challenging negatives. So as for example, the cat and dog example, what are desirable and undesirable negative pairs? So for example, we have an anchor picture of dog here. This cat image will be something, for example, is not a very good negative pair because they have very different features. They look a lot different. And then, yeah, they have different label. One is cat, one is dog. As opposed to a desirable negative pairs, for example, this one, both of the dog and cats, they are wearing the same clothes. So it could confuse the model that they have very similar features, but indeed they're actually from a different label. So those negative pairs will be more beneficial to the model training as opposed to the more trivial negative pairs. So there we tried two different sampling hard negative strategies. The first one is more intuitive. That seems to be a lot more natural. So the if anchor sequence had EC number 1111, then we'll just sample a different EC number with different from the last digit. So it will be something like 111 and X, something not 1. So basically this underlying message is saying, oh, it's a tree-like hierarchical structure, then maybe add the child node here, then that will be the most similar feature, but then it still have a different label. And the second strategy we tried is just to brute forcefully calculate the pairwise distance between the EC numbers, and then from the embedding point of view, we'll just select the negative EC numbers with the shortest distance compared with the anchor EC number. So in short, we tried both of these out, and then the second one works better, a lot better than the first one. That could be meaning the embedding doesn't really agree with the EC number hierarchy. After all, EC number is something artificial, human-defined. A tree-like structure is not naturally occurred. So once we know the sampling hard negatives, the full training loop will be something like this. We'll initialize the training process by calculating the pairwise distance matrix based on the ES1B embedding. And then from the distance matrix, we'll find the EC numbers with the shortest distance and then sample the positive and negatives accordingly. Now we train the model, and then after training the model, we'll recalculate the pairwise distance matrix so that we can dynamically find the newest hard negatives according to the current training stage. So this cycle is just training going on. And then the calculating pairwise distance and updating the new, the so-called new hard negatives are not done every epoch, but it can be a constant intervals as a hyperparameter. So after talking about the training strategy, after the model has been trained, we also need to inferences the query sequences after training. So once we have a trained model, the way to inference is pretty simple. We will first average out all the enzymes in a particular EC number. We will average out the features, the feature embeddings of all these sequences in particular EC number. And then the averaged embedding will be assigned to that EC number used as EC number embedding. So we'll do this for every single EC number. And then once we have a query sequence, we'll calculate the pairwise distance between the EC number embedding and the query sequence embedding. So now we can just find the shortest distance and then transfer the EC number label accordingly. So as a sanity check, after we train and evaluated our model, we perform an initial evaluation on a random split based on sequence identity with different threshold. So essentially 10% identity will means that the training and testing data was clustered using 10% sequence identity using threshold. And then 100% will be just untreated data set. Will be the full. And so the 100% will be just untreated UniProt database. So the comparison, the red one will be the clean result. After training, ESMB was used as a baseline to see if we're just doing this inference using ESM1B features, not instead of the clean features, how will that compare? So essentially, it's a not very rigorous ablation study to show that the contrastive learning is actually useful. So furthermore, we start to go into the result part. We start to benchmark with some of the in silico methods that developed previously, the state-of-the-art methods. We tested on 2 different independent data sets. The first independent data set comes from the SwissPro database that published like after we trained clean, so we could name it new dataset. Not very creative way, but yeah, new dataset. So these are from SwissPro, so they're human expert reviewed, kind of resemble the reliability and fidelity of the dataset. So here the other benchmark candidates are ProteinFer and DPC, 2 state-of-the-art model I mentioned before, and ECPRED, also an older machine learning-based EC number prediction tools. The benchmark metrics we chose here are precision recall and F1 score. Sorry, based on multi-class classification. And the second independent data set we chose is a very challenging one. So this one is actually a experimental study done by Price and coworkers. So it's the same Nature paper published in 2018. Essentially, what Price and other coworkers did in the paper is that they launched a high throughput experiments to annotate many or actually thousands of different enzymes or proteins in general. And then they compared with what has been automatically annotated by the database. They found out that a lot of them has conflict between their experimental results and the automatic labels given in the database, inferring that a lot of these bioinformatic-based tools or whatever tools that database uses to annotate the enzymes are not very reliable. So essentially, we picked those. We specifically picked those has been misannotated by the database. We assembled a data set. We name it Price dataset. So this data set was actually first created by ProteinFer by Sanderson and coworkers. So in this one, we also included BLASTp as a strong baseline and showing that our method can actually outperform BLASTp and ProteinFer, DPC, and other 2 more older computational methods. The reason we include BLASTp here, but not in the previous slides, is that the SwissPro database already used like bioinformatic tools to automatically annotate the label of the sequences in the new data set. Therefore, we don't want to the teacher also participate in the test. So, furthermore, we dived a little bit deeper to why Clean's performance is a little bit better than others. Especially, we think that it's because the contrastive learning can handle the underrepresented EC numbers better. It handles the data imbalance issue better. Therefore, we plotted the combined new dataset and the Price dataset based on different beans of EC numbers appearance in the SwissPro database. So for example, this 0 to 5 means that these EC numbers occurred less than or 5 times in SwissPro database. 100 to infinite will basically mean some popular EC numbers that many sequences can do. So on the left will be some rare functions, on the right will be some more popular or well-studied functions. Now, as we can see, both ProteinFer and DPC shows a bias or a clear decreasing trend as the EC occurrence decreases. As both model actually use UniProt as training data, so you can see that both model has suffered from the lack of data issue at the low EC occurrence regime, whereas clean can stay relatively uninterrupted of the EC number occurrence. So after talking about some of the in silico evaluation we have done for clean, we have also tested, performed some in vitro studies to test how clean will perform in some real life data set. So essentially, we picked halogenase, this family of enzyme, as our proof of concept or a case study. Halogenase is a perfect case study for us because halogenase is a very understudied enzyme family. It only has a couple halogenase in the UniProt database. Is also a promiscuous enzyme because depending on different halogenase you can do, fluorinase, chlorinase, or iodide or bromide. Each of them are associated with a different EC number, so they are both promiscuous and understudy functions. So it's a perfect tool to test clean in a real life scenario. Now, halogenase itself is also a very important family of enzymes used in organic synthesis because their ability to install halogen atoms to the selective substrates with very good selectivity, making them a unique unique link in part of the pharmaceutical synthesis because they can improve the selectivity for the downstream synthesis steps. Now we curated 36 different halogenases from the literature, as well as the unreviewed portion of the UniProt. And we task clean and a bunch of other different algorithms to predict on these 36 halogens that we collected from literature. As you can see, clean performs very good on this data set, followed by a cofactor, which is actually a structure-based methods to predict enzyme functions. The 4 different groups shows the different digit level of EC number. So basically to what extent or to which digit that the model could predict accurately. And across all 4 different halogenase families, the haloperoxidase, flavin-dependent halogenase, alpha-ketoglutarate-dependent, and SAM-dependent halogenase, clean can perform relatively good to at least to the 3rd level of accuracy. And from the case study of this halogenase, we take a closer look at 3 of different halogenases that clean prediction result shows some conflict with the automatically annotated results in the UniProt database. The first 2 cases are a mislabeled scenario for this MJ enzyme, and then a unlabeled scenario for the TTHA enzyme. So the MJ enzyme is, according to UniProt, it was automatically annotated as a fluorinase. And the TTHA was not able to be automatically annotated. So basically, it'll just say uncharacterized hypothetical enzyme in the database. Now we performed the in vitro experiment. So basically, we purify the enzyme, both of the enzymes. We synthesize the enzyme, purify the enzymes, and then this is the reaction we found out. They both are SAM-dependent. If they are indeed fluorinase or other halogenase, they will be able to replace this functional group with a hydrogen. Sorry, not with hydrogen. With a halogen here. But our experiment result shows that it cannot perform as a halogen. It's actually a hydrolase, so with a different EC number. So essentially here shows the HPLC result and this corresponding mass spec for the peak. the light yellow will be the SAM, which is compound 1, it's the substrate. And then the orange one, number 2, is this one, is the number 2 compound here, is a hydrolyzed SAM. Now, the green ones will be the expected location. The darker green will be for the chlorinated expected products, and then this light blue, light green one will be the fluorinated fluorinated SAM product. But neither of these two peaks have been detected. Instead, the hydrolase activity was being detected. So the HPLC and mass spec basically supported Clean's prediction, and then indeed corrected the mislabeled annotation or a unable to annotate scenario in the UniProt database. And the third of the halogens case studies, the most interesting one, is actually a promiscuous enzyme that being discovered using Clean. The enzyme name is called SSFA. So this one was only labeled as a chlorinase in the UniProt database. However, Clean gives 2 more EC number associated with it. Per Clean's prediction, it is a SAM-dependent chlorinase, and also hydrolase. So as we can see here, fluorinase will be just replacing the function group with the fluoride, and then chloride, and then this one will be a hydrolase, and then it's the same color scheme as the slides before. Number 1 will be the substrate SAM, and then number 2 will be the hydrolase product here, and then number 3 will be the fluorinated peak here. So if we added sodium fluoride, we see number 3 peak being detected, and then if we add chloride, we can see a weak number 4 peak detected as the expected chlorinase or fluorinase activity. And then again, the HPLC and mass spec result backed the Clean's prediction results. So yeah, so basically that kind of in total of the 3 different case studies demonstrated how a chlorinase can be helpful in a real world scenario on a particularly understudied and a very important, very useful family of enzymes, halogenase. So overall, we also implemented a free-to-use web server tool. So for the general public, for anyone who is not experienced in doing coding themselves, so people can just copy and paste a faster format in our website, and then click the get results. Then depending on the workload, the prediction result can be made available in a couple minutes. So if you're interested to check it out, here's the URL or the QR code that you can lead you to this web tool. So yeah, so with that, I would like to wrap up my talk today. And I would like to acknowledge, first of all, to the funding sources. NSF funded Molecular Maker Lab Institute. So we are a AI institute, one of the several NSF-based AI institute in the nation. And we're particularly interested in using AI to solve various chemistry or biology problems. And I'd like to thank my PI, Professor Zhao, for founding initial ideas and support, and all of my co-authors, Dr. Ocean Cui, who used to be a postdoc in our group, but now he is an independent PI in China, Nanjing Normal University, and Guangde for helping with the experiments, who used to be a postdoc in our group and now is an independent PI in Wayne State College, and then Dr. Yunan Luo, who is also independent PI in Georgia Tech, and also Kunal, who used to be an undergrad, worked with me, and then now is pursuing his PhD path. And yeah, and a little bit Easter egg, the cat and dog images are actually both mine. Sony is the dog, and then Jodo is the cat. They're cuddling. So yeah, that'll be everything for me. I would like to take any questions. So I think for the Q&A, should I just read out loud from the chat and then?
Yeah, or I can read it as well. And then
I think I lost track during my talk because sometimes I click the chat and then the red number will just miss. So I don't know which are the meaning.
Yeah, no worries.
Mine.
Yeah, so I can read them out in order. So
Yeah.
the first question is by Mason. Would clean work for embeddings from a different protein language model?
I'm assuming yes. We didn't use anything specific to the ESM model, so I don't think it's language model dependent. So I would think it might be able to transfer and using other language model.
Great, thanks. And then Daniel asks, how would you establish a priority list for experimental enzyme characterization to improve prediction accuracy or to discover new chemistry? Is there a native measure of novelty or surprise in the clean method?
So in this work specifically, we, if I'm understanding the question correctly, so in this work specifically, we choose halogenase because our founding institute, the Molecular Maker Institute, also has a proposal on halogenase, so it just seems natural to combine both projects and then use halogenase. Since anyways, we will be using halogenase for a different project anyways, and then it's also under the same funding umbrella. And then that's why we use it. And from a ritual synthesis point of view, halogenase is also a very repeatedly used enzyme to promote like substrate selectivities and regional selectivity and everything. So does that
Next
answer your
question.
question? I'm not sure.
Yeah, I guess also feel free to unmute if you'd like to follow up.
Hi, yeah, this is Daniel. Lovely talk. I'm just wondering, in very general sort of basic science terms, can you say that, there is an EC number missing here? Like there is a cluster of sequences, but they don't look like anything that we know. That kind of question.
Oh, okay. So essentially, if you think back of the reference process we have, we will need to have the EC number already defined in the database to get the EC number embeddings. So let me see. So I think, yeah, it's possible to be discovering some novel EC numbers if they just doesn't fit into any of the EC numbers. But I guess another limitation I have to mention here is that currently we don't distinguish between enzymes and other general proteins. So they could be just non-enzymatic activities if they don't belong to any EC numbers.
Thank you.
Yeah.
Great, thanks. Kind of related to the next question from Joe, is clean able to detect whether a protein is an enzyme at all, as well as specific EC numbers, or in other words, is there a classification category for non-enzymes?
Yes, so actually from a retrospect, we did develop a separate algorithm to give this to measure the uncertainty of the clean's prediction. So if the uncertainty is very high in the end, and then it doesn't, the distance is very long for all the different EC numbers, then yeah, we will able to characterize it as a non-enzyme. But to its nature, the way clean has been trained is really a ranking. You can interpret it as a ranking model. So it'll by all means to rank something for you. but in case if the lowest, if the top on the ranking list is still has very, very high uncertainty, then yeah, we can assume that or classify that as a non-enzyme. Yeah.
Cool. Next question from Ozza. Do you have a sense of the failure modes for clean at the various EC number digit levels?
Oh, so for the full study, we treat EC number as a label down to the 4th digit. So we haven't really done except on the halogenase data set, because halogenase data set itself, either in the literature or in the database, is a partially labeled data set. So not all the full 4 digits are available for all the halogenase. That's why we reported a digit level accuracy. But for all other scenarios, all other in silico experiments, we only consider the 4th digit as a complete EC number. So yeah, so I don't have a tested experiment to see the failure point as EC level digit.
Great. And next question is, can clean run backwards to search a database to identify all sequences that belong to a specific EC number?
I think from a user point of view, there is a way to achieve that. Maybe, for example, some enzyme function you're interested in, you can first shrink down the searching space, and then just maybe like do like a homolog search or some other tools to shrink down the searching space, and then just brute force to run the clean from the normal direction, and then you can annotate everything. But it doesn't do backwards, unfortunately.
Great. And then the next question is from Lana. Thanks for the really nice talk. For a promiscuous case study, have you ever tried other classes of enzymes, which taking very different substrates in terms of chemical structures? For example, oligosaccharidotransferase having different lipid and sugar moiety. Different halogen atoms are simple nucleophiles. Maybe my question is more about expandability. How much complex substrate can be annotated?
So just give me a sec. I missed part of the question. I'm just gonna repeat myself. Sorry. So for the in vitro experimental study, no, we only tested on halogenase. So as long as because the model itself doesn't distinguish EC numbers, right? So as long as the complex substrates or the function you're interested in was defined in some particular EC numbers, then yeah, clean can be potentially applied to identify those candidates as well. But in an experimental point of view, we only done experiments on halogenase so far.
Okay, and a few more questions left. One from Paul. Did you try other embeddings? Does contrastive learning work best with a certain type of embedding, maybe because of more or fewer features?
The short answer is no. We only tried on the language model ESM1B. So this study was initially done in 2021. So ESM1B was considered a hype and was considered as one of the best language models available. With the only thing we tweak out or the hyperparameter tweaking was the neural net itself. But no, we haven't got a chance to try other embeddings.
Great. Another question from Paul, wondering how does promiscuous prediction work? If you're assigning an EC number to the closest average to the query, does the pairwise distance need to be within a certain range for multiple EC numbers for a query to be characterized as promiscuous?
Yes. So actually, we detailed our the EC number choosing algorithm in the manuscript itself. But in short, we are trying to looking for the jump on the on a gradient on the sorted distances. So if we see there's a change on the gradient, then we'll use that as a cutoff to assign the EC number. That's one approach to assign that. The second one we can do is using p-value. So we can have a distribution of the distances. They follow a Gaussian distribution, and then the user can set a p-value, maybe like less than a certain cutoff, and then using that as a cutoff of the EC number output. Alternatively, we also have the uncertainty uncertainty evaluation model, so that depending on the user's tolerate to the uncertainty, we can use that as a criteria to output EC number as well. So it's all kind of customizable by the user.
Great, thanks. And one last question in the chat, and I think Nathan also has his hand up. Well, a question from Hani is, I'm curious if you have tried enriching the input ESM1B embeddings, or perhaps replacing with a multiple sequence alignment. In addition, the part where you average the representations from each EC seems like it could use some attention heads instead of a simple average. Has this been considered?
So we did try to enriching the ESM1B. This is after the paper published actually. So I mean, internally we try to enrich it with like structure information, but the benefit was actually pretty marginal. This actually kind of supported or agrees with what the recent paper Dr. Kevin Yang put out, that a lot of the language models are essentially another way to get the structural features, so it's kind of repetitive. Yeah, so we try to enrich with the structural features, but other than that, we try other. Yeah.
Hey, great talk. I just had a question. So my original question was, what was your motivation for using ESM1B? But you clearly answered that multiple times. But my other question was, in comparison to like a contrastive learning method, have you tried both either comparing or even combination with more adapter-based methods, essentially like freezing some of the encoder weights and then like doing extended like mass language modeling on like enzyme class of interest? Or if you're even aware of that, have you like investigated that at all?
Oh, that's a very, very nice suggestion. It's an interesting point of idea. We haven't really tried that. The reason why we turned to contrastive learning in the beginning is really the first one is we want to handle the data imbalance issue. Actually, the second is really we kind of join the similarities or resemblance from the BLAST or other bioinformatic tools, right? So if you think about it, BLAST is a alignment method. They are trying to they align the sequences, and now contrastive learning is trying to align the embeddings instead of the sequences. So it kind of feels natural to went with contrastive learning. But yeah, I think yeah, your idea is indeed very intriguing. So, but we have.
Thank you so much. Great talk.
Thanks.
Oh, one more question in the chat. Would it help to add structural information from AlphaFold or FoldSeq to the training?
Yeah, like I said before, the adding the structural information didn't really help that much. It has very marginal improvements, very incremental improvements. Yeah.
Okay, great. Thank you so much for the amazing talk. And thank you everyone for asking great questions as well.
Thank you.
And
Thank
hope
you for
to see you all
moderating.
in the next 2 weeks if there are no questions.
Thank you.